Thank our guest of today's lecture, Morgan Bomzel, head of the lab with a very long title. It's mucosal immunity and invasion and so on. So I think that Professor Margolis will present Professor Bomzel widely. So please.
Yes, thank you. So today we have a pleasure, as Ellen said, to have Professor Bomzel, our guest. And Professor Bomzel was graduated from University of Paris and then went to the famous lab in Heidelberg under directorship of very famous biophysicist K. Simons, and also worked for several years, next several years, at the United States in UCLA, University of California, Los Angeles, before returning to France and having her career in France. As Dr. Girolamo said, there are many titles, and I can continue with formal introduction, but I will stop a formal introduction. I have my own introduction, because over years of sitting in different committees, evaluating colleagues and writing reviews to the papers, I now can learn how to separate my own relations with a person, friendship, collegiality, from the objective evaluation. So objective evaluation that Morgan Bomzel, in my mind, is #1 virologist in France of her generation. And that view of mine can be supported by many, many prizes, which you can see in her CV. So it's not only my opinion, but also opinion of many committees. And I think she's getting prizes, so many that you cannot really all list them, including the prize from Academy of Science of France, some other prizes. And she became, how you call it in English, Légionnaire d'honneur. So it's the highest French order of the French Republic. And I know that this order somehow was discredited, given to some politicians who do not deserve it, but we should separate what's going to politics and going in science. It's like Nobel Prize, I think. Nobel Prize in Peace was given God knows to whom. However, Nobel Prize in Science, I don't know any scientist who got this Nobel Prize who doesn't deserve it. And I think that Morgan is one of the few people I know who are really deeply devoted to science. When I ask some of my colleagues, especially American colleagues, how is your science? They say, oh, great, we just published in this and this journal. When I ask Morgan by phone or any kind of communication, how is your science? She says, well, we did this, we discovered this. Fine, will you go to publish it? Yeah, I just accepted to PNS or to Nature Microbiology or something like this. And I think that's a really example for us how we should devote to science, and the success comes with this devotion. So I'm happy that today we have such a great scientist to teach us about her recent studies. Morgan.
So thank you for this nice introduction. And before to start, I want to thank the rector of the university and also the dean, Elena Juravieva, for this kind of invitation, and so Gloria too. And so today I will tell you about long COVID pathogenic mechanism. So as you know, so as you know, SARS-CoV-2, in 2020, the vast SARS-CoV-2 infected a large part of the world, leading to the COVID pandemic, and who affected really a lot of people. And since, also most of the people recovered, you certainly know in your environment some people who apparently recovered in the sense that they don't produce virus anymore, but that still report or suffer from a wide variety of symptoms, and that doctors don't know exactly how to treat. So these people are affected by what is known now of long COVID, and what I will tell you today about. So precisely, what is long COVID? It is an infection-associated chronic condition that occurs in patients with symptoms that last for at least 3 months after the end of the acute COVID. The pathology or the symptoms affect one of more organ systems. It can't be explained by an alternative diagnosis, and one of the problems is that there are no biomarkers as yet, and explaining why the definition of long COVID is subject to changes. The prevalence of long COVID is widely debated. It ranges from 5 to 10% and can go up to 20% or more, depending on the cohort that have been studied. But in any case, it's a lot of people if you think that almost 4 billion of people have been infected by COVID worldwide. And to give you an idea, it's a pathology that is comparable to diabetes in frequency. So clinical feature. So long COVID can affect everybody, but usually more women than men in a ratio 2 to 1. It affects adults as well as children, which is a really huge problem for them. It's independent of health, socio-economic status, and unrelated to age, sexual orientation, or ethnicity. It's also, long COVID people have been detected or exist everywhere around the world, and is independent on the geographical location. The main problem is that people affected by long COVID have impaired ability to work and for children to attend school, and long COVID can also affect profoundly emotion. So what is important, nevertheless, is that long COVID is not a diagnosis of exclusion and can be diagnosed on clinical grounds. And another important issue is that also it can resolve after months. Usually, or very often, it persists for years. And now people who were infected by COVID at the beginning of the pandemic in 2020 still have long COVID symptoms, and that's really a problem. And it's for these people, in fact, for the first waves of infected people that it's a greatest problem. So the problem in defining long COVID is due to the wide diversity of symptoms that a patient reports. And I'm sorry. And so the common symptoms are persistent fatigue, difficulty for concentration, memory changes, and all what goes together, sleep disturbance, recurring headache, also shortness of breath and cough, cardiovascular issues with a fast heart rate, too fast heart rate, and gastrointestinal symptoms. That's some of the symptoms, but up to 42 have been reported. One of the hallmark, an additional hallmark of long COVID, is that symptoms can vary dramatically between people, and also important at these symptoms, severity can fluctuate with time, and some people think that long COVID can be also, this symptom also have some cyclic appearance, and we don't know yet why. In any case, all these symptoms that have been described can be categorized in three main directions: cardiovascular symptoms, neurological symptoms, and metabolic symptoms. So, the mechanism to explain these symptoms have been proposed, and they have been proposed based on analysis of blood and tissue from long COVID patients compared to COVID recovered patients and or to healthy donor. But that's already a problem, because what is a healthy donor, especially if we want to collect a sample from healthy donor nowadays, since almost all population have been affected in one way or the other by COVID, and it's really an important issue in all these studies. An additional issue is that there is a very limited animal model, and so it's very difficult to prove this proposed mechanism. So they are mainly based on long COVID blood and tissue samples. So this mechanism range from viral persistence, immune dysregulation, microbiome dysbiosis, endothelial inflammation, neuronal inflammation, and mitochondrial dysfunction. And I will now go across all these mechanisms one by one, and to see what is known. So let's start with immune dysregulation that may increase immune reactivity. The immune dysregulation affect lymphocyte, cytokine and chemokine, autoimmunity, and mast cells. So let's start with lymphocyte. There have been observed a sustained lymphocyte activation with a decrease in naive T cell frequency, an increase in effector memory cells expressing homing receptor, that favor this memory cells migration to inflamed tissue. There is also disability of cytotoxic CD8 T cell specific for SARS-CoV-2 that express exhaustion markers such as PD-1 or CTLA4. On the other hand, B cells are also affected. They are activated and produce high level of SARS-CoV-2 specific antibody. Sorry. Probably here. As you can see here, compared to healthy donor and COVID recovered individual, here it's a scale in the log, in log scale. And this dysregulation of T cell and of CD8 of CD4 T cell and CD8 T cell, together with B cell dysregulation, induce a miscoordination between the two types of immunity and an improper crosstalk between the cellular and the humoral adaptive immunity that results in inflammation. It also suggests that there is a persistent stimulation by viral antigen, so viral antigen persistence. At the level of soluble factor, like one see that there is an increase in inflammatory cytokine, as you can see here, an increase in chemokine that induce immune cell attraction to tissue, and an increase also of VEGF that could affect endothelial cells and coagulation. And all this increase contribute to an excessive inflammation with abnormal T-cell activation, increased recruitment of immune cell, and coagulation. This study were interesting. This quantification of chemokine in this patient were interesting because it can give you the capacity to establish a score of long COVID score based on this set of these cytokines, and in addition this score so then it would be useful to differentiate people affected by long COVID and other persistent pathology, and also it could be useful to stratify long COVID severity. As I told you, B cells are activated in this patient, and no surprise then that they won't detect an increase in autoantibody, for instance, to nuclear factor, cardiolipin, however, long COVID is not associated with autoimmune disease, apart exacerbation of already present thyroiditis. Other autoantibody have been reported, such as CNS antibody against CNS adrenergic and muscarinic acetylcholine receptor, especially in patients with neurocognitive and neurological symptoms. And so it's not totally understood how these antibodies that are detected in the brain can penetrate via the brain-blood barrier, probably because the brain-blood barrier is inflamed, leaving the room to antibody to go, the junction of the brain-blood barrier being not exactly tight, leaving the possibility for this antibody to cross the. It's known that the presence of such antibody are connected to cognitive disorder, and this also gives a hint of potential therapeutical strategy. There is currently a trial with this monoclonal antibody, rituximab, which depletes antibody-producing B cell, and that may be useful for in long COVID. So as you can see, for each type of mechanism, people or MD especially have tried to elaborate a strategy to treat this patient. So lastly, for immune dysregulation, there is mast cells. As you know, mast cells are at the interface between the central nervous system and the gut and affected by gut dysbiosis. In long COVID, one observed mast cell activation syndrome that is very widely reported. This syndrome induces the release of chemical mediators such as histamine or inflammatory cytokine, and it's also mast cell activation syndrome is also known to be correlated to chronic cardiovascular, gastrointestinal, neuronal, and respiratory symptoms. And one possibility of the reason of this mast cell activation syndrome, it's the presence of cortisol that is induced by stress and factor resulting also from gut dysbiosis that could activate mast cell. And so this crosstalk between the brain and gut could contribute also to inflammation. So let's now look at endothelial cells. Endothelial cells, as you know, are at the interface of the blood and interstitial tissue and are key players in vascular homeostasis. And specialists of endothelial cells think that the main driver of long COVID is endothelial cell inflammation, as we will now see. So what is found in the blood, or what is reported in the blood of this patient, is that blood contains platelet activation marker so a wide series of these factors that are present inside platelets and released when platelets are activated. One, also the differences between healthy donor and long COVID patient in each case is statistically significant. However, the difference is never very important and remain in the range of, in a more or less normal range, even sometimes a bit increased. But what is interesting, as you these studies could be maybe analyzed in a different manner. For instance, if you see here, there is clearly a group of people that are very similar to the control, but if you would stratify this long COVID patient into two different groups, probably you will have much stronger differences between control and long COVID patient. So probably it suggests also that long COVID patient is not a very homogeneous population, but can contain different subgroup of patients. That remains to be studied, and probably the correlation between all this data and the clinical data reported by patient could be worked out much better than it is until now. Anyhow, this study indicated platelet activation and endothelial cell dysfunction. Another element that has been reported in long COVID is increased incidence in thrombosis, not only in acute COVID, but after COVID, because this increase in thrombosis lasts at least for 1 year and is associated with long COVID. Scientists looking at the plasma have detected in long COVID, but not in the same patient sampled before having SARS-CoV-2 infection, amyloid microclot that have quite large size. If you can see here, it's 20 micron is here, so it's roughly 80. The size of this microclot are roughly 80 to 100 micron. So this microclot entrap pro-inflammatory molecules secreted by activated platelets, and importantly resist fibrinolysis, reflecting a failed fibrinolytic system. So this microclot could contribute to thrombosis observed in long COVID, and some from a clinician, but especially in private company, have tried to propose to their patient to mechanically eliminate this microclot using filtering, but also this patient had to pay quite a lot of money. It was more finally a failure, and at least it was no peer-reviewed paper published at this subject. And so that's also a problem, because long COVID patients are usually easily misoriented and have to pay quite a lot of money to finally have nothing. That was one of these examples. The endothelial inflammation got quite a lot of interest, because dysregulation could be induced by a spike. Indeed, as I will show you in more detail later on, the SARS-CoV-2 spike has been detected in the plasma of long COVID patients, but not in COVID recovered or in healthy donor. And furthermore, the SARS-CoV-2 RNA, as well as the spike protein, have been detected inside lung endothelial cells. So what could do this spike? This spike can bind the receptor ACE2, as it does to infect cells. And by doing so, it not only it allows the virus to enter, which is not the case here because you don't have virus, but it can signal through ACE2, and this signalization on endothelial cells, so because this receptor is also expressed on endothelial cells, can induce endothelial damage and leakage, inducing the release of activation marker and inflammatory gene in the circulation. This, in turn, could increase endothelial cell activation marker, such as von Willebrand factor, the factor 8, and D-dimer in long COVID plasma, inducing vascular damages. And it's why all these endothelial cell specialists consider long COVID almost uniquely as a vascular disease mediated by the spike. Indeed, this sustained endothelial dysfunction would induce microvascular endotheliopathy, thrombosis in several organs, and unrestrained activation of the complement system, as we will see now. So, local complement activation have been detected in long COVID individual with the complement C5b and C6 inserting in, together with C7, inserting in the endothelial cell membrane. This induces the formation of a channel made of C8 and C9, component of the complement, forming the terminal complement complex. This channel allows thrombospondin 1, TSP1, and von Willebrand factor to escape from the endothelial, to be secreted by endothelial cells. But an additional factor is that in long COVID patients, there is a decrease in this protease, Adam TS13, which usually cleaves von Willebrand factor. And in this case, since this enzyme is decreased, the patient keep long fibers of von Willebrand factor, which C3b so C3b, the member of the complement, keep associated with the von Willebrand factor. And also there is coagulation factor is increased. The coagulation factor 11 is increased in long COVID. So this long fiber of von Willebrand capture easily monocyte activated monocyte forming conjugate with activated platelets and start to provoke coagulation. And in addition, the monocyte together, so this conjugate between platelets and monocyte allow the release of thrombin, which will activate C3b, which in turn, via the alternate pathway of the complement, will in an autocrine feedback loop reboost the complement activation system, and propagating this endotheliopathy. So the crosstalk, altogether the crosstalk between complement and coagulation machinery at the endothelial cell surface certainly contribute to thrombosis, which is one of the issues in long COVID patients. So let's look now at mitochondrial dysfunction, which may impair metabolism and lead to fatigue and metabolic symptoms. So it's known that the long COVID patients have intolerance to exercise, and in a cardiopulmonary exercise test, one have detected an increase in lactate production as well as a decrease in fatty acid oxidation. This sign suggests mitochondrial dysfunction and perturbation of mitophagy, the mechanism that remove unfunctional mitochondria in cells. Other people have analyzed, or other scientists have analyzed blood by mass spectrometry and to study metabolites. And as you can see here, in red is long COVID, in blue COVID recovered, and in gray, so healthy donor. Long COVID differ widely from the two other populations. At the level of metabolite detected, precise metabolite detected, there is an increase in non-saturated fatty acid and a decrease in carboxylate, polyamine, and tryptophan. And pathway analysis shows that in long COVID patients there is dysfunction of fatty acid metabolism and mitochondrial dependent lipid catabolism. This element indicate a clear metabolomic signature of an altered mitochondrial respiration. So then other scientists have published studies looking directly at the mitochondrial respiration, but this time in a time-resolved manner, as well as they study reactive oxygen species and mitochondrial DNA, which all contribute to mitochondrial respiration. This study has been performed in monocytes, especially of patients with cardiovascular complications. And as a result, scientists reported a blunted mitochondrial respiration with decrease in ATP production, decrease in ROS species, and depletion of mitochondrial DNA from the cell, and maybe a secretion, so either a total destruction of this mitochondrial DNA, or maybe a secretion of this mitochondrial DNA that can then in turn bind to TLRs on other cells and propagate inflammation. So persistent bioenergetic failure, impaired oxidative stress, and mitochondrial damage in these cells could contribute to cardiovascular, or is known to contribute from other study to cardiovascular symptoms, and could be here one of the reasons could here contribute to long COVID symptoms. Again, this study can give some potential hint to therapeutical strategy. One could treat patients with antioxidant, mitochondrial repair agent, or metabolic modulators. So what about microbiome dysbiosis? So as you know, microbiota and the brain communicate bidirectionally via various routes, such as the immune system, the tryptophan metabolism, the vagus nerves that connect that vagus nerve, and the enteric nervous system. This communication between the brain and the microbiota affect also all peripheral neurons, and therefore all organs innervated by peripheral neurons. The microbiota gut organ axis is influenced by genetic and epigenetics, of course, but also the way of life. And then next, this has an effect on food intake, cognitive behavior, stress, source of stress, social interaction, and also fear, all symptoms that have been reported by long COVID patients. So therefore, there have been some studies on to characterize the gut microbiota changes in long COVID patients, and all the study agrees together that one observes an increase of gut bacteria that induce inflammation and endocrine disruption, and a decrease in the good bacteria that perform immune homeostasis. And thus, one can think that via the gut organ axis, this microbiota could induce, could affect the organ that are innervated by peripheral neurons, and in turn induce the symptom reported by COVID patient. Again, that could be used for therapeutical strategy. One can use microbiome-based therapeutic using like symbiotics, probiotics, for instance, that would allow a restoration of the proper microbiota, and thus hopefully reduce fatigue. And such, so you can do that by fecal transplantation, and there have been some attempts to do that, and indeed some of the patient goes better. So let's then now finish this description by focusing on viral persistent. We think that is a master, or could be the master regulator of all the other symptoms. Indeed, viral persistence, so as you know, virus can persist in the body, such as HIV, which is a retrovirus, also RNA virus, but retrovirus, and DNA virus, which makes sense, such as, because it's quite stable, such as hepatitis B, CMV, or EBV. However, viral persistence also has been reported for a wide variety of viruses, such as Zika, or the Japanese encephalitis, West Nile, hepatitis A, the polio, so from measles viruses also, so for many different families of viruses. And interestingly, some of these pathologies listed here are very similar to one or more symptoms reported by long COVID patients. So it's not a stupid idea to search for long COVID, for SARS-CoV-2 persistence in long COVID. So as I told you before, the first evidence of viral persistence is the continuous detection of spike and RNA in the circulation of long COVID patients up to several years, not just months. Here I give you so that we have found in the lab too. And here there is just two examples. Here in the plasma there is RNA and spike, 12 up to 12 months post-infection, and here in the plasma, a spike in the plasma up to 14 months post-infection. But the level of the spike reported by these various studies is very variable, from 1 picogram to 1 nanogram per ml, so 1,000-fold differences, and also, importantly, variable in frequency of patient. That can vary from 5 to 60%. So that remains a problem to understand well the role or the impact of this spike in the circulation, and might be due to different technical issues, and also the control used to define what is positive or what is negative, especially between COVID recovered individual and long COVID patient. So in any case, so we think, so our results based on 100 patients compared to 30 COVID recovered patients also point to a frequency more around 20%. And in any case, in all these studies, there have been no clear correlation between the presence of the spike in the plasma of this patient and specific symptoms. The only thing which has been reported is a correlation between spike in the plasma and the number of long COVID symptoms reported, which doesn't help really to understand the mechanism. So, and that's especially for Lonia, and spike has also been found associated to extracellular vesicles in long COVID patients, but I'm not specialist, and I leave that to him to explain to you the meaning and how it goes there. I don't know anything about extracellular vesicles. So now looking in tissue, many studies have reported SARS-CoV-2 RNA and protein in various autopsy tissue from patients. For instance, here, that's an example of spike and its RNA. Here's a spike and here's a viral RNA detected in bronchial tissue 3 months post-infection. Or here, another study, 3 months post-infection, in neuron in the cervical spinal cord, here in green the spike and the neuron in violet. Other study reports the presence, not the spike, but also the nucleocapsid and its RNA, here in macrophages, in the appendix, up to 14 months post-infection, or here in the gut mucosa, 7 months post-infection, and here in this study, which these values are not always provided, between 20 to 45% of the patients were really positive for nucleocapsid and its RNA. So the mechanism behind the viral persistence remain poorly understood we can discuss about if you want later, but it's quite an interesting issue, many different hypotheses, but until now, even from where, what is the origin of the spike found in the circulation? How come a spike, which is supposedly a transmembrane protein, can be released in the circulation? Where does it come from? Where is a reservoir? Or where do the virus persist? And one can imagine that to get in the circulation, an enzyme has to cleave this protein, but it's not known exactly from where it comes from. Additionally, all these studies have shown up to now measured or observed, quantified viral component in these tissues. But it's not known whether all these components correspond to actual viruses that are produced, and if these viruses are infectious, and if this is the case, can the infection be propagated from one site of replication to another? And also, I have to recall you that SARS-CoV-2 is not a circulating virus. It's produced in the mucosa, but it's not detected in large quantity in the blood. So, all this question is the question we have investigated in the lab using platelets as a model. So, why platelets? It's because we have performed studies in severe acute COVID-19 and so during the pandemic, and found that the megakaryocytes, which are the cells in the bone marrow, theoretically at least, that produce platelets by thrombopoiesis. And we found that megakaryocytes in the bone marrow are infected by SARS-CoV-2 in this severe acute COVID individual, and also megakaryocytes were found infected in the lung. Indeed, relatively recent studies demonstrated that megakaryocytes are not just present in the bone marrow and in the lung. And in fact, just for the story, the presence of megakaryocytes in the lung has already been described almost 100 years ago, and, but it has not been much studied apart 10 years ago in a mouse model where they could show that in the mouse, which is not exactly the case in the human, 50% of the platelets come from megakaryocytes located in the lung. So, nevertheless, in severe acute COVID, megakaryocytes are infected by SARS-CoV-2, and this megakaryocyte infected by SARS-CoV-2 during thrombopoiesis, so here you have proplatelets and then platelets, platelets capture viruses in vesicles as well as viral RNA. And these platelets, we found that this could demonstrate that these platelets were sheltered spike as well as the positive strand of viral RNA. And here the platelets, for instance, here the platelets is detected by an antibody against its surface in green, CD41. You can see that inside the platelets, not at its surface, one can detect RNA, and in this case, the replicative form of viral RNA. We could also show that these platelets contain virus that was infectious for macrophages and could spread infection. We then show that infected platelets are mostly detected in COVID non-survivor, people who passed away 2 weeks after collection of the sample, but also in a small proportion of patients who survived COVID. And recently we have found a question this survivor with SARS-CoV-2 in platelets, and they reported to all report, so small number, but they all report to have developed symptoms compatible with long COVID. We therefore decided therefore hypothesized that SARS-CoV-2 persists in megakaryocytes and platelets, forming a replication-competent reservoir which could contribute to long COVID symptoms. So for this study, we collected blood from long COVID patients and compared to COVID-recovered individuals, that is, people having had COVID and having no symptoms 3 weeks after infection, so having properly recovered. So to summarize, we found that megakaryocytes and platelets in this patient shelter the viral spike, here in green, here it's megakaryocyte, and here the spike, double-strand RNA, which is a replicative form of the viral RNA, so suggesting that these megakaryocytes indeed produce viruses. And here you can see platelets that also contain viruses and RNA, both positive and negative strand. Here I show the spike, which is inside the platelets. The platelets is labeled in red, and here by confocal microscopy you can detect the spike only inside the platelets, not at its surface, with a frequency of platelets containing viruses to 0.1 to 2%. But now, if you consider that blood contains 150 to 410 to the 9 platelets by liter, it's quite a lot. It's roughly 1% of platelets is infected. So that's 1%, if you take the mean of this frequency, correspond roughly to 10 to the 9 infected platelets by liter of blood, which is at the end not too low. So this study suggests that megakaryocyte is one, at least, of the viral reservoirs that could produce viruses. So the next important question was to ask whether this virus found in platelets is infectious. And for that, it's indeed what we found. Using a reporter cell system, we found that SARS-CoV-2 in platelet is infectious for reporter cells. And importantly, so you may know that platelets end their life in macrophages. And we could show that, it's not astonishing that platelets interact with macrophages. And we could show that platelets from long COVID patients containing viruses can transmit infection to macrophages, suggesting that platelets not only contain viruses, but are Trojan horses to spread infection. And finally, we also found that in this patient, platelets from microaggregates, as you can see here, that's platelets in COVID-recovered individual, and in long COVID patients, these red aggregate are aggregate of 50 to 100 platelets together. And we also measure mitochondrial respiration of this platelet and could find that it is reduced in favor of ATP produced by glycolysis, indicating that platelets could contribute, as I showed you or explained to you before, to immunothrombosis. So altogether, we think that viral persistence is really at the origin or an important factor that could drive all the other symptoms reported in long COVID. So let's start with viral antigens, which are detected. they can affect the adaptive immunity, virus-specific B and T cell, which then would activate modulate lymphocyte activation, T cell exhaustion, antiviral antibody secretion, inflammatory cytokine secretion, and complement activation. In other side, viral RNA could, via toll-like receptors, for instance, could affect the innate immunity, especially myeloid cell, and in turn autoimmunity, latent virus reactivation, phagocytosis, impaired phagocytosis and antigen presentation, and inflammatory, so also have a role in inflammatory cytokine secretion. So clearly there is an uncoordinated crosstalk between these two adaptive and innate immunity, leading to immune activation that in turn can affect mitochondrial dysfunction, microbiome dysbiosis, the gut nervous system axis, neuronal inflammation, what I told you about, microglial inflammation, also the hypothalamus pituitary adrenal axis dysregulation, and the endothelial dysfunction. And in turn, this would provoke the metabolic symptom, neurological symptom, and cardiovascular symptom described or reported by long COVID individuals. So, what remains to do? A lot, because as I told you, all these mechanisms are proposed based on analysis of blood and tissue, and that's not extremely dynamic, apart maybe for respiration that can be studied in real time, but otherwise a bit difficult. And also, the problem is these pathological mechanisms are not predictive of any specific symptom in individual patients. And I think a lot could be done by working more on the correlation between all these measurement, biological measurement, and specific clinical data reported by patients. And models should be refined, and it's also what we are starting to do in the lab, trying to group symptoms by groups to see if a group of symptoms correlate more or less with this or that. An interesting axis is also to evaluate whether the symptoms reported are really specific, and mechanisms are specific for long COVID, or some may be common with other acute infectious pathology. Chronic infection produced by influenza, chikungunya, or dengue, as I mentioned before. So not only viruses, but also bacteria, such as tuberculosis or Lyme disease, Borrelia, Coxiella, and also with non-infectious pathology that share a lot of symptoms with long COVID, the MECSF. There is also a need of biomarker and a need of course, specific treatment, but as I mentioned throughout the talk, some trials are currently made by different groups and may give some hope to these patients for whom, for now, nothing really can be proposed. So I think I stop here, and so just acknowledge people from now. And take your question.
Of course, maybe students first, since thank you for your brilliant presentation, a lot of new information for me personally. So maybe first question from students.
So it was very interesting, I'm gonna say, and we had a case of that, and there is a patient and most of the symptoms you discussed met. And my question is like, how dangerous is it, and if there are any death cases, because doctors cannot really do anything in this case, as we know. And yeah, it's been a long time, and I'm quite worried about that person as well, besides my scientific interest. So yeah, how dangerous really it is.
So dangerous, so it's not, so people will not die from long COVID. But the problem is that, indeed, so that's one of the reasons I'm working on long COVID, and because I have a niece that says 20 and affected by long COVID, and there you start to. And when you go to this meeting and listen, especially for parents of these young, even children that are affected and can't exercise and can't go to school, you start to think you have to do something. And I think the only thing we can do as scientists for now is to try to understand how it works, what is behind that, because that's also something very difficult. The field is controlled essentially by MDs, so it's clinical research. They have patients, they have symptoms. But okay, a lot are declarative, which is also one of the problems. What do you measure? So they pretend they exclude other pathology, clinicians exclude other pathology, but still, how do you define the gravity of these symptoms? You can declare symptoms, but your symptom the person you know may have, how does it compare to the symptom another person has? All this declarative information is very difficult. So I think the best that can be done is what some scientists started to do. You have seen a lot of data here, measuring things. And I think what really the people try, so scientists run for, is to have a biomarker. Because if you have a biomarker, which is really clearly associated with long COVID and not with long Lyme or long influenza or long something else, that would help a lot. So it's why, in one sense, the score that is built on inflammatory cytokines is quite interesting. It has been proposed by a lab in the US, by the lab of Patterson. The problem, once again, is that guy has a lab in the university, but he has also a company. And then, okay, he tried to sell his score from the side of his company. So it has been criticized, also it makes sense, and other people try to repeat to see if it's true. But the only thing you can do at this level is to try to understand the pathology. And for virologists, I'm not exactly a virologist, but I think what is fascinating in the basic science point of view is the story of persistence of an RNA virus. Because RNA is a quite fragile molecule. How come in the cytosol of a cell, not only platelets, but of a cell, the RNA is not destroyed? And how can it persist for so long? So many hypotheses you may have heard about lipidless organelles and other, so the way SARS-CoV-2 also replicates involved large production of membranes, and the virus, the RNA may be kept there, protected there. So that's a field, I think, fun. As in one sense, HIV helped the study on HIV for all these years helps to understand many things, not only in virology but also in immunology. I think focusing on viral persistence in SARS-CoV-2 might be something very interesting. Not sure I'm answering your question, by the way.
Thank you. I have some questions regarding this. My first question is, what is the current level treatment for long COVID? Is there any drug treatment or similar therapies? And my question, another question is there another treatment that could potentially completely cure this long COVID symptoms and clear out, like targeted, cell therapy that could, stem cells that could regenerate, for example, mitochondria dysfunctions or endothelial cells, or inflammation processes using, for example, antioxidant therapy. And my third question is, what are the virus factors that could contribute to these inflammatory pathways of such that results these inflammations specifically in the endothelial cells and mitochondrial dysfunction? Thank you.
There's a lot of questions. I think the first was about antivirals, if I'm correct. The treatment. So one could think, so treatment, as I told you, a lot of things are in progress. There is no real treatment. That's really a problem. That's why we need to understand, before to have a treatment, we need to understand the mechanism. And then we can address each mechanism. But I think the most important is to target viral replication, but there are no antiviral specific for SARS-CoV-2. All what has been used are usually antivirals that have been recycled, especially for HIV. That works not extremely well. And in fact, during the COVID pandemic, several antivirals have been given to patients, but with not much success. So people have tried to use this antiviral for long COVID people, but it doesn't work, so it's probably not the specific antiviral, and that is a field that should be worked out. The second is about, so it's what I think should be, the research should be focused on, really, if indeed viral persistence is a master regulator of all these symptoms, I think that should be targeted. Then you asked me about all these options, all I went through during the talk, that's possible, antioxidants, but so in fact patients read a lot of general literature, but even quite specialized literature, and then take antioxidant, all options that are suggested from the observation made, but it helps, it can help some, not others, and doesn't cure. The problem is to cure them. That help, like eating in one way and not the other, like, so many things have been proposed and tested by patients, but none of them really cure them. We have, for instance, a trial using with clinician colleagues using Maraviroc to see if that it's a ligand of CCR5 that is, so CCR5 being known to be involved in the gut dysfunction. For now, on the 10 or something patients we have included and studied, it works for some, Maraviroc works for some, but not for all. And that's also an issue. So I really think before to try to imagine a treatment, that's maybe the bias because I'm a basic scientist, you have to understand the mechanism. Or then by chance, as many other drugs have been discovered over the year, penicillin and whatever else, by chance, maybe one day somebody will come out due to a mistake, due to with a treatment. But for now, I think a rational way to establish a treatment has to go first through understanding of the mechanism.
Yeah. Thank you for a very interesting presentation. I have several questions. The first will be, if there are symptoms you described found in people what we call health, had no previous history of the infection. Second question would be if there is any correlation between these long COVID symptoms and the vaccination. And the third one would be, we heard about the many symptoms associated to long COVID. If there is any discrimination between these symptoms, which are the primary and which secondary, because that would be interesting to know for therapeutic approaches.
So
that's all.
Yes. So very good question. So to start with vaccination. So long COVID occurs in both vaccinated and unvaccinated individuals, and to complicate the system, clinicians come to us telling us that some of their patients pretend to have declared long COVID upon vaccination. So it's very confusing, but for now, on the vast majority of the patient, so it's half-half. Some have been vaccinated once, twice, 3 times with whatever mRNA vaccine or attenuated vaccine, the same. The one vaccinated, not vaccinated, develop the same way long COVID. What was the first question?
If the similar symptoms are found in
Health people,
which did not have the previous COVID infection. So you said that prevalence was roughly 50% in the whole population,
yes.
so the half did not get the COVID. So if the similar symptoms are found in, let's say, healthy people in terms of COVID.
Yes, of course. It's an extremely good question, and I think that's also something which is fascinating and start to be studied not just for COVID but for other diseases. The history of patient and the capacity of a first infection, previous infection, to modify the immune system or even the cell at the not at the genetic level but the epigenetic level. And this mechanism is called trained immunity. That's a field that's blooming now. So it's known that once you get infected, you may keep a type of innate memory due to these epigenetic changes induced by the first infection. And one second, you are subject to a second infection, totally disrelated from the first one. You can another infectious diseases. Your body remember and react much faster than if you didn't get this first infection. So this trained immunity mechanism so has been very much studied with the BCG, with the vaccine against tuberculosis, which has been shown to induce epigenetic mechanism. But it occurs also after other viral infection and bacterial infection. And on this line, so this train immunity mechanism pushed by Romanian scientist but working in the Netherlands, Michael Netea, he tried during COVID to vaccinate people with BCG to see if they will be protected from long COVID, from acute COVID. And that worked more or less. It was not a full success, but he got reasonably good results. So the same for persistent infection. So this strain immunity, these epigenetic marks could last. It's very variable again, from several days to several weeks to several months, not much more than 6-7 months, maybe 1 year. But that, I think, the history of the disease of the patient is certainly very one of the way to go to understand the differences between these patients and between people who recovered and people who didn't, who developed long COVID. I think that's a very good question.
Yeah, just a small comment on previous question. So it would be nice to see these kind of people, if symptoms in healthy people present, as additional control when comparing different parameters between healthy COVID and long COVID. Additionally, healthy people with the same symptoms. And the third question was, we saw the huge amount of the symptoms associated with long COVID, 42 symptoms, if I am correct, associated with different organ systems. And I wondered if it's possible to discriminate the primary symptoms and then secondary symptoms caused by primary symptoms. So it was the whole net of interaction between.
It's an interaction. Sometimes the follow-up is people who didn't recover. It's not so, and some appear, but usually it's people who were. But also it was mild COVID. But then they can't recover and start to develop these symptoms. But we don't have much. So I think then it's very difficult, because one of the issues in understand long COVID is that individuals, so humans, are not mice. And then it's very, so you have to rely on declarative information. And if you take somebody who is very ill now, very depressed, you ask him what was his symptom during acute COVID, how do you know it's true or not? And it depends how you ask the question, who asks the question, and then also the collection of clinical symptoms is something really very difficult to manage. So that's a problem. I think it's a good question, but unless you have an animal model where you can control objectively what is there, it will be extremely difficult. And again, symptom. You know, to be tired. What does it mean to be tired? How do you handle tiredness? Depends on the people.
Thank you. And my question is about this, that you mentioned in the beginning that there was any relation between age or ethnicity or sex and so on. So have you excluded in your data set, let's say, people with chronic disease or people who get other infections besides COVID? And if you compare these target groups, for example?
That's true that at least I can tell you for our cohort, part of these people have other disease, but not all of them. And if you take if you try to summarize, so to make meta-analysis of what is published, nothing comes out.
Okay, thank you.
But it's always very difficult. Yes?
Yeah, I have two questions, short questions. One for the beginning of your lecture, one for the end. So one is related to the previous, probably. So there are 42 symptoms. When you listed them, I'm sure that all of us found some of them in ourselves. Fatigue, difficult to concentrate, or something like this. So, coming from mathematics long ago, is a definition. Do you need all 42 symptoms? Do you need 2 of them? Do you need 1 of them? How you define long COVID for the first time?
That's very, so I think so. Several symptoms. In our cohort, we consider 3.
But there should be a definition. Yeah, I need 5 symptoms to diagnose, or 25.
That's a problem to be on the declarative.
Yeah, that's
relying, symptoms relying on declarative information.
Yeah, but very difficult.
And because what might be done now more regularly is to give not only, to ask people not only to declare symptoms, but to provide them with list, ask them to grade the symptoms.
Yes, that I understand, but it's difficult to study disease which is not well defined.
But so that here we are logical scientists. We have to deal with clinicians. Clinicians are logical a bit, rational a bit, but they have to deal with patients. And they have, I have discussed with a clinician who is also a basic scientist from Canada, and who was asked by the Social Insurance of Canada to evaluate people that pure clinicians declare to have long COVID. And he looked, he interrogated his patient, and he said, I can't measure anything for now. However, I know that they are not okay, and all what they declare is true. And it's not due to pathology we know, it's something different. It's all the problem of chronic disease.
Right.
It's like, so all chronic disease.
I remember there was such a disease as chronic fatigue, and people even found herpes virus 6 as a etiological agent before they found there is no such a disease.
Yes, so it's why this cytoscan score.
Yes.
What I told you, and it's I think written here, should have written maybe bigger.
Yes, and for
need a biomarker.
Understand, need for biomarker.
Definitively a biomarker specific for long COVID,
yes,
and not for something else.
And
cytokine may be a hope.
Right.
I can tell you, because I will not tell you what, we found neurotoxin produced by this individual, which looks like very discriminating.
And my second question for the interview, so you very convincingly showed us that there is virus in platelets, or viral RNA in platelets, which may of course explain the whole thing, as your hypothesis states, but I still don't understand, because half-life of platelets is a week, and the same about megakaryocytes. So how it can persist for a year if the platelets already gone, which initially infected, megakaryocytes also gone.
I think you may want to come to my lecture tomorrow.
I will, I promise.
And so, as I told you, so platelets are a transitory shelter of all these viral elements, but they are produced by megakaryocytes. So megakaryocytes in the bone marrow and in the lung have a longer half-life, but still not extremely long. So probably the real reservoir is progenitors of megakaryocytes. And so people from my lab who left and have a new lab start to work on that. Don't forget that it was the same story with HIV persistence, where we and others found that megakaryocytes are infected. We found that they are persistently infected, and a group in the US reported that precursor of megakaryocytes is a precursor of megakaryocytes that are infected by HIV. Yes, stem cells. But I don't see how it could be used, how it can be used for a cure. It's probably the reservoir. But it's very difficult to access. So you can make model, you can start from iPSC and try to infect them, but it's a bit complicated to get
thank you
model once to get this.
Thank you for interesting lecture. I want to ask you if you exclude the patients which already were vaccinated from your experience, or you include all type vaccinated, or all of them?
Because there is no real difference. It's not really, there is no correlation between vaccination
and I also have a question about this. You mentioned that this RNA that should be digested still remaining there inside this cell. And have you ever seen this kind of situation in another type of the virus or any type of the RNA? Or this is the first time when the RNA is not degraded and still remaining there?
So we didn't work really on other viral pathology apart HIV. And in HIV, as just mentioned, we find megakaryocytes persistently infected despite efficient antiretroviral treatment. And megakaryocytes still, so but that here is a retrovirus, so the RNA has been retrotransferred and inserted into the host cell DNA. Yes. And then replicates the virus.
Yeah, but also in HIV, in this case, it was first go back to the DNA and then remain in the megakaryocyte in this state, right? But in this case, you found that this is RNA that are remaining inside the cell. Or you think that maybe it reverse transcriptase is for HIV as it's for HIV, or what?
But I'm not exactly sure. So
okay. So my question is that you found that RNA is remaining for a long time inside the cell, right? But you mentioned that in HIV case it was the same situation, but in HIV it is because it's reverse transcribed into the DNA state and then translated into the RNA, and that's why it is remaining there. But do you think that the same situation is for this virus, or it is just the RNA that stay and remain? But
so what we show is that this RNA part of it can produce, so replicate and produce viruses. So viruses are obligate intracellular pathogens, so they need or they make use of the cell machinery to produce viruses. However, as you say, the RNA could remain there, and also part of this RNA could bind some toll-like receptors and contribute to inflammation. So both, but more studied. So I think it's both, in fact. I don't see why.
Just like the idea on treatment, can we use antisense nucleotide to the spike gene for megakaryocytes or not?
Why not? But then you have to target all. You have to target them in vivo.
Just two short questions. So first of all, gut microbiome dysbiosis. Do you think that this is the result of the viral infection, or maybe in some cases it can be a result of antibiotic usage during the treatment in the hospitals or something like that? So is this result specific for the COVID virus, or it's more common for the treatment at hospitals, or in some cases at home also? This is the first one.
In fact, I don't work on this topic at all. It's just by reasoning. Long COVID occurs a lot in patients who have not been in the hospital. So in mild COVID, so they had COVID. So they were not treated for additional pathology with antibiotics. So I don't think that. And what is very interesting is that all studies looking at microbiome, gut microbiome, all say the same. So even if it's different cohort, it's not always comparable. So I would say it's induced by, it's correlated with viral infection. Who start? Probably background also. And as you know, it's very difficult, even if you pretend that you will change the intestinal flora, it's extremely difficult.
And another point you have mentioned, the changes in the lipid metabolism, mitochondrial metabolism of lipids. Did you check what is, for example, in case of blood cells in progenitor cells or megakaryocytes or something like that? So, and can we connect it with the diabetes then, for example, or something like that?
Possible,
Thank you.
but nothing has been published on this topic. So I want to say, so most part of what I told you, it's not my study at all.
I was just curious about, you mentioned that there are some challenges regarding the mechanism behind everything, how SARS-CoV-2 life cycle is in the human organisms or systems. And the second challenge was to detect this pathogen SARS-CoV-2. And have you ever wondered that you can use computational tools such as AI, or there are new tools are being developed, it's called AGI, to just study the whole processes, model them, and discover biochemistry or something, the mechanisms or hypothesis-based models or modeling them, and then it can be tested through clinically or experimentally. That's my question.
It's very good strategy. You should have a lab. Indeed, so I think the models that have been used, and so the correlation between symptoms and biological measurements that have been made until now are too simple. So just a bioinformatician arrived to the lab a month ago, and we tried to modelize in a more complex manner, different manner. And what you can do, so you know much better than me clearly, you can use the data, train your algorithm with part of the data, and then validate your algorithm with the remaining data to see if it's true. Because always the problem, you can make models, but how do you know it's true? So I think there is probably a potential to using AI for sure, but you need specialists, and not only bioinformatician, but people who know also the biology. That's always a problem in modelization, because for now you have all this omics data in all directions, and then people don't know the disease enough, and then propose mechanism, but it's too general. You need to focus on, if you want to find drugs, you need to be more precise. But I think, yes, of course, make use of AI for sure. That's maybe you should do that. Because now a lot of data are available, all the omic data are available through publication in website. You have to deposit. And by the way, for looking at megakaryocyte in acute COVID, we also, and their transcriptomic can show that they have emergency there is an emergency megakaryopoiesis. We made use of data, omic data available publicly. It's very good.
I think we can discuss more and more, but you are tired, and also I think that this topic was really actual, not only for Georgia, but for the whole world. Thank you again. Also, I just want to announce tomorrow, Professor Morgan Bomzell will have lecture with medical students on the platelets. So if you want to join medical students, you also can attend this lecture tomorrow at 3 o'clock in the same hall. So thank you again. I think that huge information, not only for biologists, but also for the physicians, is just very small souvenir in French from the Ilia State University, and I hope we will host you again, maybe next year also, and maybe we just organize some joint seminar with several professors in immunology and virology. Thank you again.